So we're really thrilled and honoured to be able to end this meeting with a lecture by Sir John Gurdon. So, as you know, John performed a very famous series of experiments that revealed the potency of the differentiated cell. And I think that when you learn about these experiments, perhaps as an undergraduate, one thing very quickly becomes clear, which is that the only possible course of action is to spend your life as a developmental biologist. And I know that many of you, like me, have really been inspired and motivated by John's work. And indeed, he's continued to work over many years to understand the mechanism by which cells can become reprogrammed. So from amongst his very many awards and honours, I'm just going to very quickly highlight three. So we have an institute in Cambridge named for John, the Gurdon Institute, where many of you work. In 2012, he was awarded the Nobel Prize. And furthermore, the BSDB Undergraduate Summer Studentship Scheme is named after John, so the Gurdon Awards. And we were really delighted a couple of years ago to be able to award a Gurdon Award to an undergraduate to work with John Gurdon in the Gurdon Institute. So, and that just really highlights his ongoing support and training for the next generation of developmental biologists. So what better person to close this special meeting? And John, we look forward to your talk.
This is the subject I want to talk about, and I do get a fair number of invitations to speak, not related at all to any scientific contribution I may have been considered to make, but almost entirely to my school report, which the gist of which is that I was thought to be interested in doing science, but the master said that if I can't learn simple biological facts, there would be no chance at all of doing the required work, and it would be a total waste of time on my part and those who'd have to teach him. So that was the end of my scientific education at school. Now, what I'll try and cover three subjects in this talk. One is the reprogramming of differentiated cells back to an embryonic state, then the stability of differentiation or resistance to reprogramming, and then a bit at the end about the prospects for cell replacement. So we have to go back to the 1950s and remember that the original question at that time asks whether all cells of the body have the same genes. It's hard to remember that we didn't know that at that time. And the great pioneers in this field were Briggs and King, who discovered how to transplant a living nucleus to an egg. That was a great discovery, particularly Robert Briggs, and at my age I have the privilege of having known these great people. Unfortunately for him, he worked on an unfavorable species, Rana pipiens, and that his work showed that when you transplant nuclei from mature embryonic cells, they no longer survive. Now, I came in at that point, and I have a distinguished scientific ancestry, starting with Boveri, whose student was Spemann, whose student was Balzer, and his student was Hadorn, and my own supervisor, Michael Fischberg, came after that, and I'm one of the et ceteras after that. Now, this is my supervisor, Michael Fischberg, and he took me on kindly, and I had two possibilities or a possibility what to do for a PhD, and this illustrates perhaps an interesting general principle. The position is that the key experiment which I was invited to investigate had already been done. So you might say, why put a student onto a project which has already been completed? And the result was that when Briggs transplanted nuclei from post-gastrulus stages, there was no survival. So that left me with two possibilities. Either to repeat the experiment done and possibly hope to get a different result and maybe even a different conclusion. Or if that didn't work, it would be worth trying to find out what the mechanisms are in trying to make nuclear transfer work and why it did not initially give any significant survival. So that's where I started, and I was lucky that it worked out, and it means that if a student is given a project that seems to have no hope at all, that doesn't actually mean to say you won't have a bit of luck and get something back out of it. So Michael Fischberg made early on an immensely important discovery, which was the one nucleolus genetic marker, which was necessary for following the fate of transplanted nuclei. Again, there's an interesting principle there. He had a student who was working on haploidy, the one set of chromosomes, and haploid embryos do not survive. However, this student found that when you recognize haploids by a single nucleolus, which you can see on the left, they survive perfectly well. And if I'd been the supervisor, I would have said, well, you better just see if you can repeat that experiment, remake your solutions and check everything and do it again. And she would never have been able to do that. But he didn't. He said, you have to go through the collection of xenopus that we have in the laboratory and see if you can find the actual one female which laid the eggs which gave this surprising result. And amazingly, that worked. So he found there was, in fact, a particular mutant discovered by chance whose offspring gave this one nucleolus offspring as a diploid embryos. And that was a crucial result which was immensely helpful in this project. Now, it wasn't entirely straightforward even then, because when we tried to inject a needle into these eggs, they have this enormous jelly round which made them essentially unpenetrable. And if you made a pipette and pushed it into the egg, it just came out the other end with all the jelly around it. So that was a major problem, and we found, perhaps by luck, that if you ultraviolet irradiate the egg, that dissolves the jelly and, by good chance, also kills the egg nucleus. Now, the other technical problem was to make a needle of the right design, and in those days you had to make these things, you didn't buy them, and we found we could make a microforge so that you could shape the tip of this needle, which was normally broken off randomly, into a really sharp end, rather like a hypodermic syringe. So that meant we could go ahead with experiments, and I'll now just review the evidence that we had for nuclear transfer reprogramming. And I was lucky that as a graduate student, I got the first adult cloned animal, and this was a very healthy frog. It lived for about 20 years and had about 4,000 offspring, so it was a really good animal, and there was no reason to think that cloning has any bad effect. Now, that led to the possibility of using specialized cells as nuclear donors. So here's a muscle cell. You can see the muscle fiber going through, and you transplant that nucleus into the enucleated, unfertilized eggs. When you do that, as often happens when you use nuclei of specialized cells, the early embryo is abnormal, and it dies within 24 hours. However, if you take the part of that embryo which has normal-looking cells and retransplant a nucleus from one of those into another set of eggs, then you get a fair amount of survival, including in cases like this with a nuclear transplant compared to a fertilized egg, and the eyes of those nuclear transplant tadpoles derived from a muscle nucleus were essentially normal with all the right components. So it illustrated that there really had been a major reprogramming of the transplanted nucleus, even from a specialized cell. Now, the conclusion from that bit of the work was essentially this, that the egg is designed to transform sperm to an active embryo nucleus. And the materials of the egg try to do the same for a transplanted nucleus. On the other hand, the nucleus is designed to maintain expression like differentiated cells do, and so it is designed to resist change. So it's a kind of battle between the recipient egg cytoplasm and the nucleus. So concluding this early phase of work, the main point was the answer to the initial question, which is it seems that cells, or nearly all cells in the body, have a complete genome. If it weren't for that, then it wouldn't be possible to use the technique for reprogramming. The egg cytoplasm has a remarkable ability to reprogram gene expression back to an embryonic state. And lastly, as cells differentiate, their nuclei become increasingly resistant to reprogramming by eggs. So that left two questions. What are the components of eggs and oocytes that can reprogram somatic cell nuclei? And the second question was what causes the resistance of somatic cell nuclei to being reprogrammed? And these are the two remaining questions that I deal with in the rest of this talk. So the first finding was that within a remarkably short time of transplanting nuclei, there is a huge exchange of the so-called linker histones. In this case, we were transplanting nuclei marked with the linker histone in GFP into the recipients, and the recipients were supplied with the same linker histone marked with GFP. And I hope this will show a video of the exchange. You should see that almost all the green nuclei have now become red. They've taken up the linker histone, which is special to the egg. The two actual green marked nuclei are interesting. They're ones where the membrane of the donor cell had not been broken. And it makes the point that when you do any nuclear transfer, you have to have exposure of the cytoplasm of the donor cell to that of the egg. Otherwise, you don't get any reprogramming. And so to summarize a large amount of work, we can see the following events happen. Here is the early exchange of the linker histone taking place in about 2 hours. And then you get histonate 3.3 replacing the linker histone, again after about 4 or 5 hours. Then there is the major demethylation of chromatin, DNA and protein. Then you have, still within a day, you have actin polymerization. That's probably related to the mobilization of the nucleosomes. And finally, the elongating polymerase II starts to actually act within less than 24 hours. So there's a very rapid, huge change in the chromatin after nuclear transfer. And looking at it diagrammatically, you can see that you have the nucleosomes compacted, the linker histone comes in and opens them up, and then the histonate 3.3 replaces the histone B4, as it's called, and finally the polymerase II, the 2-serine kind, comes in and starts to transcribe nuclei. So let me now turn to what I regard as one of the really interesting questions in the field, and this relates to the stability of cell differentiation. The position is that most of our cells do not change within our life. They have 70, 80 or so years, and they retain the same level of differentiation or commitment to it. If we could really understand that, that might help us stabilize cell differentiation and stop the cells going off the intended track. So I go back now to one of the first, I think, major experiments. This is Ernst Herder, and he did this, in my view, wonderful experiment transplanting imaginal discs from a larva. He'd take it out, divide it into two, transplant half into a young larva, let it grow up, and then when it had grown to the late larval stage, and it was still an imaginal disc, he would take it out, divide it into half, and do it again. And eventually, he would let this larva go through metamorphosis with ecdysone and form a fly, at which point the imaginal disc cells revealed what their memory was. And if you start with a wing disc and you get it back there, you would find that in almost all cases, the cells remember what they were. Now, he was able to do this for some 50 generations. It's about 500 cell divisions, and it's an amazing example of the enormous stability of cells in a progenitor state. That was, I think, a great experiment. My own colleague, Eva Hermann Seder, has taken this on to analyze epigenetic memory in nuclear transfers, and you might say, what is epigenetic memory? Now, if we take this example where you have endoderm cells with an endoderm-expressed gene, strongly expressed, and you then compare this expression to what would be true of the ectoderm cells of a fertilized embryo, and of course this endoderm-specific gene is now not expressed. In the case of memory, you transplant the endoderm cell nuclei and grow them up and look at the ectoderm where that gene should not be expressed, and you find it's now strongly expressed. So that means that even after nuclear transfer, there is a memory of the previous state of that donor nucleus. Now, to summarize a large amount of this work, this, first of all, illustrates that as you go later in donor stage, these are the stages of the embryos, but as you go later, you find that the success rate of the nuclear transfers goes right down, as Briggs and King found themselves. If you do this demethylation, H3K4, you now improve the success rate substantially, like twofold, almost back to the level of a fertilized egg by that demethylation. So it gives you the demonstration that this modification is a critical part of the memory of the transplanted nuclei, and in order to make these work, you have to remove the memory to get success. So I now turn over to another kind of experiment which is important in analyzing what is happening. For this purpose, we use nuclear transfer to oocytes, and when you do the normal nuclear transfer experiment, you transplant a nucleus into the egg, and there is a massive replication of DNA for about 10 divisions, during which time the chromosome replication is often abnormal. And many of the abnormal results of nuclear transfer, somatic cell nuclear transfer, result from the damage to the DNA during this enforced replication. However, if you transplant nuclei, multiple nuclei, into the germinal vesicle, as it's called, or nucleus of the oocyte is in the first meiotic prophase, there is no DNA replication, no damage to DNA, and the very same nuclei persist for a few days. And this multiple nuclear transfer to oocytes is a valuable system because you get very rapid response, no DNA replication, no cell division, many nuclei per injected cell, and you can do these experiments with mammalian cell nuclei. They show this quite remarkable reprogramming. This is the Xenopus oocyte and the huge germinal vesicle. And the germinal vesicle is critical in these experiments. And you may know that the amphibian oocyte nucleus has about 1,000 extrachromosomal nucleoli with the highly transcribed ribosomal genes. So this nucleus has extrachromosomal loops, circles of DNA, which are intensely transcribed. What we do is to inject nuclei into this environment of the heavily transcribed DNA. And when you do that, you find that the pluripotency markers, SOX2 or NANOG, are turned on within 1 or 2 days, almost immediately, very rapid activation. So that enables one to ask, what is the component of this germinal vesicle, which is itself needed for normal development? And we're dealing here with about 95% efficiency instead of the usual 2%. So now my colleagues have examined this in some detail, and the main result now is that if you look at SOX2 transcription, which is one of the pluripotency genes in the transplanted ES or fibroblast nuclei, this is increased by 100-fold in 2 days. It's an enormous effect. And the other genes, which the non-pluripotency ones, are not changed in that way. So that, for example, SOX2 and OCT4 are not turned on this time scale if you transplant myocyte nuclei into the oocyte. And the mechanism of this dramatic reprogramming depends very largely on histone exchanges and on histone or DNA demethylation. So we're now turning over to another question, and this has to do with the mode of action of transcription factors. Now, you may recall that throughout development, one is always calling upon transcription factors to direct cells to their new fate. They're key components of cell lineage determination. And the current view of the mode of action, which is not in detail known of transcription factors in general, is that they have a very rapid dynamic effect. They almost always are considered to dissociate from the DNA which they bind to in a matter of seconds. Now, we've wanted to use this system of oocytes to examine whether this apparently dynamic binding and unbinding of transcription factors is in fact generally true. And the experiment we have used for this is the following. We inject messenger RNA into the cytoplasm of the oocyte, and then you give it a day, and the protein encoded by the messenger RNA concentrates in the germinal vesicle. So the proteins are made, and they home in on the germinal vesicle. Now, we then can use plasmid DNA as a competitor and this is all done with the ASCL neurogenic transcription factor, in order to see whether this binding of the ASCL to its DNA can be competed off. That's a really specific binding competition experiment, which is very difficult to do in any other system, like with transfected cells. And so you flood the system with the competitor protein, and then you assay. And the result, oh, by the way, this works extraordinarily well. So if you have messenger RNA in such a system with DNA, you get these enormous counts on the luminometer. with a background of about 50, and if you miss out the message, it's still down many hundredfold on the effect that you see. So it's a very strong assay for the effect of the factor. And the end result of this, which has been going on for a long time, is that the competitor cannot compete. So you have and this is on the log scale, so you get a very strong response in the way that I've described. But if you supply competitor at hugely increasing amounts, it makes almost no difference to the removal of the protein from the DNA. So it seems that there is an extraordinarily long dwell time, at least with these lineage-determining factors, and our longer-term view is if we could find out why this is, what are the cofactors which stabilize the binding of a transcription factor, that could help us understand the general stability of cell differentiation and possibly increase that stability in some beneficial way. So that's the result of this, and it makes the point there is a clear distinction between dwell time, the amount of time a protein stays on DNA, and the site occupation time, which is the time for which the responding site has the factor in it. So concluding this, a transcription factor as bound to DNA, initiated transcription, it seems in this case to be extraordinarily stable, and we conclude that a cofactor or cofactors are needed for this stability. And this concludes the part that I'm going to talk about of our own work, and these are the people who've helped me, so I acknowledge them, particularly Jerome and Eva. In this case, I'm quoting the work of Peter Coffey and Lyndon da Cruz, who do this work. And the position is if you suffer from macular degeneration, you lose central vision, and eventually you lose all vision completely. And this is a relatively common disorder for elderly people. Now, the position here is that the photoreceptor cells connect with the retinal pigmented epithelium cells, which remove debris and which promote the survival of the rods and cones of the photoreceptors. And what they can do is to convert cells like fibroblasts or other cells into a sheet of cells on a piece of plastic, and these can be converted into the retinal pigmented epithelium cells. And this material can be grafted into the back of the human eye if you're as skillful as Lyndon da Cruz is, and you get some substantial rescue. And there was a recent case in the newspapers where their work has progressed very well in patients. The reason why this seems to work for the vision in humans is that the cell number is small. So a human heart will have 10 to the 11 cells. The retinal pigmented epithelium of the eye is only about 10 to the 4 cells. So moderate number of cells is very helpful. And you also need cells of a given type. So these are retinal pigmented epithelium cells. And the position now is when I talk about this, people say, well, that's wonderful. When can I have this treatment? And the answer is you can't. So why is it that when an advance like this has taken place, you cannot have it? And the regulatory bodies control this, and they will even not allow a patient to sign away their rights because they say you have to have informed consent. So the position is that this, I think, amazingly hopeful treatment for a large number of people is still about 4 or 5 years away. And so the question we come to now is who takes these decisions? Why is it that you're not allowed to have this replacement even in one eye to test it out? And really the question is who takes decisions? Is it lawyers, priests, politicians, or even patients? I would hope that eventually laws will change, and it's the patients who can say that they understand what is done and they would like the treatment. And there are many, many people who I know would very happily opt for this outcome. So the summary of this talk is the following. It says that nearly all cells of the body have the same genes. Nuclear transfer can reverse differentiation by chromatin components, or eggs or oocytes, and the IPS mechanism turns out to be completely different. It doesn't depend on chromatin exchange. The stability of cell differentiation is promoted by stable transcription factor binding, and in my view, the cell replacement therapy has, I think, good prospects for small cell numbers and single cell types resulting from this whole field of nuclear reprogramming. So finally, I end up with one comment going back a while, even before I was born, to Confucius. And he made many famous statements, one of which I really warmed to, and that was this. He says, if you really like the work you are doing, you never need to work another day for the rest of your life because it doesn't seem like work. And that's where I've enjoyed a great lot of help over my career, and I'm grateful for that. Thank you.